My name is Devrim Gozuacik. We are working in Sabanci University, Istanbul, Turkey, on regulation of autophagy in mammals. We are focusing on different proteins, pathways, RNAs regulating autophagy. There are several techniques to study autophagy. We are using molecular biology, biochemistry, and cell biology techniques to analyze different pathways that we are discovering. We are using immunoprecipitation, immunofluorescence techniques, confocal microscopy, and various other techniques to analyze autophagy pathways. Details of different techniques and pitfalls and troubleshooting will be explained by my PhD student, Secil Erbil.
The overall goal of immunofluorescence and immunoprecipitation procedures is to monitor endogenous protein-protein interactions in a cell. This method can help you to answer the key questions about protein-protein interactions and the cellular localization of them. The main advantage of these techniques is the opportunity to quantify the interaction intensity during desired treatments and conditions in comparison to control. To begin, detach the cells with trypsin. Then, count the cells using a hemocytometer and calculate the cell concentration. Take packed and autoclaved cover slides and place them in a 10 cm petri dish. If you are working with an easily detaching cell line, such as HEC293T like in this experiment, coat the cover slides with sterile filtered 0.01% polyallyl lysine solution.